and this reverse rotation is called recovery stroke. So the rotation from first position to second position is called power stroke, and the movement from second position to the initial stage or from final position to initial position is called recovery stroke. And this power stroke and recovery stroke will continue, and this whole process is resulted in the formation of muscle contraction. We are explaining the muscle contraction by sliding filament theory. Sliding filament theory. The name that is the sliding filament theory is given by or due to, we know that here we can observe the movement of actin filament. So due to the movement of this actin filament, that is why we are calling it as sliding filament theory. The name that is sliding filament theory was given, or it is due to the movement of actin filament. Due to movement of actin filament. So what is happening here? First of all, we are discussing the whole step of muscle contraction. The first one is relaxed stage. This is the first stage. Here we are taking a Z line, actin filament and myosin. In actin filament we can find some active sites and which are covered by troponin. So there is no connection between actin and myosin, that is no actin myosin cross bridge here. This is the relaxed stage or first stage. Then next one is stimulated stage. In stimulated stage or during stimulated stage, the muscle fiber will receive stimulus. So due to this, the receival of this stimulus, the T tubules will release calcium ions, and this calcium ion will combine with the troponin present in active site of actin, because the troponin has higher affinity towards calcium. Due to the combination between troponin and calcium, the active site of actin is now free, and to this free site the myosin head, here we can find there will be a myosin head. To this active site the myosin head will attach by using energy produced during one molecule of ATP hydrolyze. We should note that a small amount of energy from this whole part is using here, and the remaining energy is using for the rotation or a power stroke of the myosin head. Then during next stage we are taking the here the myosin head is attached with the actin filament, and due to the energy released during one molecule of ATP hydrolyze, the myosin head is rotating from one position to second position. Here we are taking first and second, from first position to second position, and this rotation is called power stroke, and this power stroke taking place in head part of myosin. So by this power stroke the myosin head will pull actin filaments in towards the center. If the actin filament is moving, then the Z line will also moving, because this Z line is associated with the actin filament. Then we should note that there is no movement for myosin here, only the actin filament and the Z line are moving. Then after some time this will regain its original position, and this one is called recovery stroke. And this continuous process, that is power stroke and recovery stroke, resulted in muscle contraction. And we are explaining the muscle contraction by using sliding filament theory. Here we can observe the movement of actin filament. here we can observe the movement of actin filament. That is why we are calling it as sliding filament theory, because the actin is moving or sliding here. Okay, then so these are all about muscle contraction. Then we are taking the sarcomere. Sarcomere. What do you mean by sarcomere? Sarcomere is the distance between two Z line. If the consider if there is a Z line here, then the portion between two Z line is called sarcomere. During this muscle contraction, we can observe that the Z line is moving towards the center. So during muscle contraction, the length of sarcomere is decreasing. Length of sarcomere is decreasing. Then we are considering another part, that is H band. What about H band? H band is the distance between two actin filament. This is the H band, distance between two actin filament, and we can observe that due to the movement of actin filaments towards the center, the length of H band is decreased. Length of H band is decreased. Then we are taking next, that is A band. What about A band? A band is the length of myosin, that is from the length from one end to another. This one is the A band. We can observe that there is no movement for A band here, only actin and Z line are moving here, so there is no change in length of A band. So A band remain same. So during muscle contraction, the length of sarcomere decreasing, the length of H band decreasing, but the length of A band remaining same.